In this chapter, we're going to discuss carbohydrate metabolism. This is a general outline for the chapter on carbohydrate metabolism. We'll first talk briefly about digestion and absorption of carbohydrates. We'll then spend some time talking about the chemical reactions in glycolysis to produce pyruvate. Pyruvate then is the molecule that gets converted into acetyl-CoA and then enters the citric acid cycle to produce NADH and FADH2 that enters into the electron transport chain to produce energy. We'll also talk about glycogen and how we can degrade glycogen so that it can enter the glycolysis cycle. Then we'll talk about glyconeogenesis, some of the terms used for glucose metabolic pathways, and finally, we'll talk about pentose sugars and how they enter into the glycolysis pathway. Let's look at some first-level metabolic pathways for carbohydrates, lipids, and proteins. As you can see here in the green, those are our dietary lipids. They enter to the metabolic pathways to form glycerol and fatty acids. Fatty acids then can go through a cyclic combination to form acetyl-CoA. Proteins, they undergo the first levels of digestion to form amino acids. At that phase, they enter in through cycles, including the urea cycle to form urea. But in addition, they can enter into the keto acid metabolism chain, which will eventually go to form pyruvate and acetyl-CoA. What we're going to talk about in this chapter are the carbohydrate metabolic pathways. In this case, we're going to talk about going from dietary carbohydrates through digestion to form glucose, through the glycolysis process to form pyruvate, which goes on to combine with coenzyme A to form acetyl-CoA. There is also a reverse process where I take pyruvate and recombine it to form glucose. We call that gluconeogenesis. So for the glycolysis process, we take food, we digest that food both in the mouth, stomach, small and large intestines to form glucose molecules, which then undergo chemical processes to form pyruvate. That would be the glycolysis process. And eventually, pyruvate goes on to form acetyl-CoA. If I look at this molecule, this is an alpha keto acid. Here is the carboxylic acid side of the molecule. On the alpha carbon, there is a carbonyl group or a ketone. This is an alpha keto acid. So the process by taking glucose to make pyruvate is called glycolysis. The process by taking pyruvate and turning it back into glucose is called gluconeogenesis. Gluco standing for glucose, neo meaning new, genesis meaning let's make some. Digestion is the process of breaking down food molecules, cleaving bonds, by hydrolysis reactions into simpler chemical units. For example, if I take starch, which is a polysaccharide, and I do hydrolysis reactions at each of these bonds right here, each of these glycosidic linkages, I can make molecules like the disaccharide maltose and some other small polysaccharides. This occurs in the mouth using the enzyme alpha amylase. It catalyzes the hydrolysis of alpha-1,4 glycosidic linkages in starch and in glycogen to produce smaller polysaccharides and disaccharides like maltose. However, limited carbohydrate digestion occurs in the mouth because food is usually swallowed quickly. In the stomach, very little carbohydrates are digested. That's because once the enzymes enter the stomach, like amylase being swallowed, it gets deactivated or it gets denatured by the acidity of the stomach, so it's no longer an active enzyme. Most of the carbohydrates that we eat are digested in the small intestine in areas called intestinal villi, which are areas with very high surface area. In those intestinal villi, there is an enzyme called pancreatic alpha amylase, which hydrolyzes the remaining polysaccharides to the disaccharide maltose. So I've gone from polysaccharides to disaccharides. The final step occurs within of digestion of carbohydrates occurs in the outer membranes of these intestinal mucosal cells to form monosaccharides. These monosaccharides are then absorbed by the intestinal wall in the villi and are actively transported to the bloodstream and onto the liver for metabolism. One of the main processes of glucose metabolism is the glycolysis process. So that is the metabolic pathway in which glucose is converted to two molecules of pyruvate, one ATP molecule, and one NADH molecule. And most of this takes place in the liver in the cytosol. If we look at these metabolic pathways, here is the pathway I'm showing here. Glucose through the glycolysis process to pyruvate. This is a 10-step linear process where I have glucose, one chemical reaction, 2, 3, 4, 5, 6, 7, 8, 9, 10 to form pyruvate. Pyruvate then is ultimately transformed into acetyl-CoA, so it can enter into the citric acid cycle onto the electron transport chain to make ATP, or pyruvate can go by a reverse process called glyconeogenesis to reproduce glucose. We'll talk about the glycolysis process and each chemical reaction involved in transforming glucose into two pyruvate molecules. The first step is a phosphorylation process where we take glucose and we add a phosphorus molecule to the C6 carbon. This is an energy expensive process in that it takes an ATP molecule to produce that glucose 6-phosphate molecule. The second step is an isomerization reaction, converting glucose 6-phosphate into fructose 6-phosphate. Notice here I have a pyranose ring, 6-membered heterocyclic ring. Here I have a furanose ring here. If I count carbons here, 1, 2, 3, 4, 5, 6 carbons. I've maintained all my carbons. 1, 2, 3, 4, 5, 6 carbons. I've just used an enzyme to change the structure from a pyranose ring into a furanose ring. The third step in the glycolysis process is another phosphorylation step. In this case, I'm taking fructose 6-phosphate, using the phosphorus off an ATP molecule to put a second phosphate group on there to form fructose 1,6-biphosphate. Also notice that this is an energy expensive process in that I use one ATP molecule to form an ADP molecule. The fourth step in the glycolysis process is to take the 6-carbon fructose 1,6-biphosphate, here shown in the open chain form, through the use of aldolase enzyme to form two new 3-carbon molecules. One is dihydroxyacetone phosphate. The other one is glyceraldehyde 3-phosphate. The fifth step in the glycolysis process is to convert one of the two 3-carbon molecules into the other. In this case, we're going to take the dihydroxyacetone in the presence of an isomerase enzyme and convert it also into glyceraldehyde 3-phosphate. In other words, what we have here now is we have two molecules of glyceraldehyde 3-phosphate. The sixth step is an oxidative phosphorylation step where I take my two molecules of glyceraldehyde 3-phosphate in the presence of NAD+ and a phosphate molecule to form the 1,3-biphosphoglycerate. Notice in this case, I've generated an NADH molecule in this process because if I oxidize something, I need to reduce something. I produce a molecule which can then go on to the electron transport chain and produce energy. Step 7 in the glycolysis process is to transfer a phosphate group from my 1,3-biphosphoglycerate molecule over to the ATP molecule to form the energy molecule ATP, so I'm gaining energy here. My final product in this step is the 3-phosphoglycerate, so I only have one phosphate group now on that molecule. Remember that I have two of these molecules that are moving through the glycolysis chain, so I produce two ATP molecules and I produce two 3-phosphoglycerate molecules. Step 8 in the glycolysis process is an isomerization reaction where I take my 3-phosphoglycerate molecule and I move the phosphate group from the third carbon here down to the second carbon here to form a 2-phosphoglycerate molecule. Step 9 is a dehydration reaction where we remove the hydrogen and the hydroxyl group at carbons 2 and 3 to form the phosphoenol pyruvate molecule. Notice here I still have a phosphate group attached to the molecule. The final step in glycolysis is to remove that phosphate group from my phosphoenol pyruvate, and in the process I put a phosphorus group on ADP to form ATP and my pyruvate molecule. Note again that I have two AT molecules that are produced because I'm moving two 3-carbon molecules through the glycolysis process. Let's now summarize both the ATP consumption and the ATP production during glycolysis. In step number 1, we needed to use an energy molecule ATP for that chemical reaction to occur. We also needed ATP energy in step number 3. In step number 6, we produced two NADH molecules because we now have two of these 3-carbon molecules moving through the rest of the glycolysis process. I can then take two ADP molecules. I can phosphorylate those to form two ATPs in step 7. And also in step 10, I take two ADP molecules through a phosphorylation process to form two ATP molecules. Doing the mathematics, I have a net gain of plus 2 ATP molecules. So pyruvate is then converted to acetyl-CoA using a specific enzyme, which then enters into the citric acid cycle to produce more ATP. The same thing happens with NADH. It enters into the electron transfer chain to produce more ATP. Let's look at the fate of pyruvate under oxygen-rich conditions, in other words, aerobic conditions. Pyruvate will react with coenzyme A in the presence of NADH to form acetyl-CoA. Notice here I've started with a 3-carbon pyruvate. Now I have a 2-carbon acetyl group attached to my CoA molecule. I've oxidized this molecule up to CO2. If I oxidize something, I need to reduce something. NAD plus acts as my oxidizing agent. It gets reduced to NADH, which then can go on to the electron transport chain and produce ATP. So the purpose of pyruvate in aerobic conditions is to maximize energy output by making ATP molecules. In other words, I convert pyruvate into acetyl-CoA, which enters the citric acid cycle, and it pushes NADH and FADH2 into the electron transport chain to produce ATP. Let's now look at the total number of ATP molecules produced per glucose molecule. In the glycolysis process, there are 2 phosphorylation steps which both consume an ATP molecule, so I have a net loss of 2 ATP molecules. I also produce 2 NADH molecules during the chemical reaction to convert glyceraldehyde 3-phosphate into 1,3-biphosphoglycerate. That NADH enters into the electron transport chain to produce ATP. I also create ATP directly in the glycolysis process during my dephosphorylation steps because I have 2 of these chemical reactions occurring with each of the 3 carbon molecules. I produce 4 ATP molecules directly during glycolysis. When we oxidize 2 pyruvate molecules to 2 acetyl-CoA molecules, each of those reactions produces an NADH molecule, which can enter into the electron transport chain. If we look at the citric acid cycle here, we produce 2 NADH molecules in this step, 2 more in this second step. We produce a third molecule called GTP, which is guanosine triphosphate, which acts just like an ATP molecule, but instead of having adenine, it has the guanosine nitrogenous base. I also, in the process of these last 2 chemical reactions here, each of them produces 2 FADH molecules and 2 NADH molecules. All of these NADHs and FADH2 molecules then enter into the electron transport chain. For each of the NADH molecules from glycolysis up here, I produce 1.5 ATP molecules, in other words, 3 total ATPs. For each of the NADH molecules formed in the oxidation of pyruvate, I produce 2.5 ATP molecules, or 5 total. For each FADH formed in the citric acid cycle, which there are 2, each of them produces 1.5 ATP molecules, or 3. And for the 6 NADH molecules formed in the citric acid cycle, each of those NADH molecules is used to produce 2.5 ATP molecules for 15. Adding them all up, 1 glucose molecule through the metabolic processes yields 30 ATP molecules, which then can be used to do other chemical reactions. It's the energy source for life. So far, we have talked about the fate of glucose moving through the metabolic pathways to form pyruvate. What about other carbohydrates like fructose, galactose, and the polysaccharide glycogen? Those can also enter into the glycolysis process to form pyruvate. Let's take a look at each of these 3 different molecules as sort of representative cases how we can take monosaccharides and polysaccharides and they can be metabolized into the glycolysis process. Both galactose and fructose are converted to intermediate products that can enter the glycolysis pathway directly. For example, if I look at galactose, I can use an ATP molecule to add a phosphate to galactose to form glucose 1-phosphate. Glucose 1-phosphate is a molecule that is already in the glycolysis process, so it can enter in directly. Fructose enters in by a several-step chemical reaction where I take fructose, I react it with an ATP molecule to form ADP. In other words, I'm forming fructose 1-phosphate. Another chemical reaction turns that into glyceraldehyde. Another chemical reaction reacts with another ATP molecule to form 2 glyceraldehyde 3-phosphates. They can enter into the glycolysis process right here at step number 5. Glycogen, which is a polysaccharide used by animals to store glucose molecules, it is broken down in a 2-step process called glycogenolysis to form glucose 6-phosphate. So I take glycogen, which is a long polymer of glucose, here N representing the number of glucose molecules, that is reacted with a phosphate molecule in the presence of an enzyme to cleave off one of the glucose molecules. So now I have N-1 for my polymer, but I create a glucose 1-phosphate. That glucose 1-phosphate then goes through an isomerization reaction to form glucose 6-phosphate, and then it can enter into the glycolysis process right here between steps 2 and steps 3. This slide summarizes the glycogenolysis process, where we start with glycogen. It is a polymer of glucose molecules. In this case, it has N+1 glucose molecules to make up glycogen. That glycogen molecule reacts with the glycogen phosphorylase enzyme to add a phosphate group to the end glucose molecule to generate glycogen -1 glucose molecule, so just to the end. That end glucose molecule is cleaved to form glucose 1-phosphate. It then reacts with another enzyme to form glucose 6-phosphate, which can enter directly into the glycolysis process. That glucose 6-phosphate can also react with an enzyme called glucose 6-phosphatase to form glucose, which again is our energy molecule, which can then enter directly into the glycolysis process. Notice that these last 2 steps here are reversible steps. So I can take glucose, react it with the enzyme hexokinase in the presence of an ATP molecule to form glucose 6-phosphate, which can then isomerize back to glucose 1-phosphate. And it can also react with a uridine diphosphate molecule to form a uridine diphosphate glucose complex. I then take a glycogen molecule in there with the enzyme glycogen synthase to form my glycogen N+1. So this whole cycle here, I can take glucose molecules and put them together to form glycogen, keep on forming glycogen. When the body needs energy, I can take those glycogen molecules and start creating glucose 6-phosphate and glucose, which both can enter into the glycolysis process to form energy. Let's look at some of those chemical reactions in the glycogenesis process a little closer. If I start with glucose 6-phosphate, I can do an isomerization reaction to move the phosphate group from carbon number 6 over to carbon number 1. I can then react that glucose 1-phosphate molecule with the uridine phosphate molecule to form this new molecule called uridine diphosphate glucose molecule. The third step is to take my UDP glucose molecule, react it with my glycogen chain, which is just a series of glucose molecules in the presence of an enzyme, to form a growing chain of glucose molecules, adding one glucose unit. In that process, I produce a UDP molecule. I need to convert that UDP back into UTP. I do that by reacting it with the ATP molecule, which is generated in the electron transport chain. So in that case, I generate a UTP molecule, which can go back to create a UDP and glucose molecule. This ADP molecule then has to go back into the electron transport chain to create an ATP molecule. So to add one glucose unit into glycogen, that requires the investment of two ATP molecules. One to take glucose and form the glucose 6-phosphate molecule, and the second one to regenerate the UTP molecule. The opposite pathway to glycolysis is called gluconeogenesis, where I take molecules like pyruvate and make them back into glucose. The process is not exactly the opposite of glycolysis, but there are many similarities, and we'll talk about a few of those in the next slide. The roles of gluconeogenesis are multifold, but two of them are the brain requires glucose all the time, and if we didn't have an intake of glucose, we need to start changing pyruvate into glucose, or we need to start converting glycogen into glucose. Also, glyconeogenesis helps maintain a normal blood level at all times, especially when we have inadequate dietary carbohydrate intake. In other words, we can no longer do glycolysis. This following diagram shows the interrelationships of the different glucose pathways. For example, if I start with glucose, I then can form glucose 6-phosphate, which can go through the glycolysis process to make pyruvate. I have pyruvate, I can go through the gluconeogenesis process to reform glucose 6-phosphate, which can go on to form glucose, or it can go on to the glycogenesis process to start forming glycogen. I have glycogen, which is a storage type of polysaccharide, which can then undergo glycogenolysis to reform glucose 6-phosphate, which can then form glucose again. Also, if you look at this diagram here, there are several other pathways that pyruvate can take. The one we've talked about a lot so far is to make acetyl-CoA, which can then enter into the citric acid cycle. Two other pathways which are reversible. Pyruvate can go on to make lactate, or it can go on to make ethanol. Let's look at those two alternate pathways for pyruvate. Under short-term anaerobic conditions, in other words, oxygen-poor conditions, for example, during heavy exercise in the muscles, I take a pyruvate molecule, I react it with an NADH molecule and a proton to form lactate and NAD+. So the purpose of this chemical reaction is to produce NAD+, which can then be used in the acetyl-CoA and the citric acid cycle to produce energy. Later on, after the aerobic conditions are reestablished, meaning I have oxygen now, the Cori cycle can take lactate and make it back into pyruvate. So muscle fatigue associated with strenuous physical activity has been attributed to the increase of the buildup of lactate in the muscles. Some microorganisms have the ability that under anaerobic conditions they can convert pyruvate into ethanol and carbon dioxide. For example, here I take pyruvate, react it with an NADH molecule and 2 protons. In a two-step process, I produce ethanol and NAD+. I can use that NAD+ in the glycolysis process, and I can also enter it into the citric acid cycle to produce NADH, and it can go on into the electron transport chain to produce ATP. So this involves two reactions here. One to remove one carbon from that pyruvate molecule to form acetaldehyde, which is a 2-carbon molecule. And then finally, I reduce that aldehyde to the alcohol to produce ethanol. We see this all the time when we're using yeast to produce either leavening in bread or alcoholic beverages. And that ends the discussion on carbohydrate metabolism. In the final chapter in this course, we're going to be looking at lipid and protein metabolism.